So you said you spent 10 years in Japan. So do you know how to say zebrafish in Japanese?
Fish in Japanese is sakana.
Okay.
Okay. Yeah.
Zebra sakana. How many percentage of human genome and fish genome are quite the same?
75 to 80%.
What diseases can be studied and has been studied using zebrafish?
To answer your question, there is hardly any disease that has not been modeled in fish yet. Almost every disease has been modeled.
Even Parkinson's.
Even Parkinson's, even cocaine addiction
Wow.
has been modeled in fish.
What is the response? Even they're addicted to cocaine?
One of the most widely used product from marine source is the green fluorescence protein, which is obtained from jellyfish.
Which was it? Oh, jellyfish, right.
And it is the most important protein in cancer research. In India, of course, zebrafish is emerging as an important model system. I'll tell you why it has become popular, or rather why it has gained more popularity in recent years.
Hey guys, welcome to QED podcast. The following is the conversation with Dr. Anirban Chakraborty. He is a researcher, educator, presently serving as a director at NUCSER, Nitte University Centre for Science, Education and Research.
That's right.
Okay. So welcome to the podcast, sir.
Thank you,
Great to
Clinton.
have you here.
Thank you very much. Thanks for having me. Yeah.
Yes. So today's conversation is going to be about a very special fish. It's called zebrafish. And Anirban sir has been working over decades, more than a decade, over this fish.
Yes.
So I want to understand what is so special about this fish and what has been cooking at Nitte
Right.
using zebrafish. And I'm pretty curious about it. So let's get started.
All right.
Okay.
So first of all, thank you, Clinton, for coming over and taking up such an exciting topic, which is very close to my heart.
Okay.
I perhaps know a little bit more about zebrafish than you. So I will try to explain what the model is, what the fish is all about. And yes, as you rightly said, what's cooking with fish? Unfortunately, we can't cook this fish. We do, there is a possibility to cook it, but then we can't eat it. It's a very small fish.
But
So
there's
this-
a fishy business in Nitte.
Not
Something
everything is
fishy.
fishy. There are good things as well. So the zebrafish, basically, it's a model system. It's a vertebrate model system.
What do you mean by vertebrate model system?
Okay, I'll come to that.
Okay.
So model systems are basically in vivo systems which can help you study about human diseases or study about effects of certain toxicants
Without involving humans.
without involving humans, because our ethics do not permit us to do experiments on human beings. So we resort to animal systems. They may be prokaryotic system or eukaryotic system. They may be vertebrate system or they may be invertebrate system. They may be multicellular or unicellular. So we use these animal systems to study the consequences of certain toxicants, pollutants, certain genetic alterations, and of course, effect of certain infectious agents on the physiology, on the general functioning of the fundamental processes in the body. And what we learn from these animal systems most likely would give us an idea what would probably be happening in a human system if a human being were to be infected or having a genetic alteration in their genome. So there are many, many different model systems that you know about, right from the most simplest one is called E. coli. Escherichia coli, which is a bacteria basically. This is a prokaryotic system. A lot of major discoveries in the field of genetic engineering has happened through this simple bacteria. And it is one of the most indispensable organism in biotechnology. You cannot have a biotechnology without an E. coli getting involved in it.
Right.
So we have E. coli, which is very simplest one, and then we have all the way till mouse or rat,
Right.
which are the mammalian model systems that we know about. Zebrafish comes somewhere in between. The zebrafish is basically a vertebrate system, but it is not a mammalian system. It's a non-mammalian vertebrate system, originally identified or originally studied to know the developmental biology of fish.
Interesting.
But then, somewhere in 90s, a very famous scientist in Germany, Steininger, his name is, he understood the basic developmental aspects of this fish in great detail. And then he wrote a book called the Zebrafish book, which basically tells you how to raise this fish in lab conditions, how to identify different developmental stages, and how to work on the various stages, and basically how to use this model system for developmental biology. And then came its major breakthrough discovery is its utility in studying biomedical science. So biomedical science, basically application of biology in medicine.
Which year was this where they found use of zebrafish in biomedical science?
There are
Is it
plenty
pretty
of
recent?
universities around the globe, but I would say Max Planck in Germany, they are the pioneers. In studying the effects of certain mutagenic agents on genomoid effect on zebrafish.
Right.
And they ended up identifying quite a few mutants.
But which year was it? Like
Way back in the 1990s.
1990s.
1990s, yeah, way back. So, since then, zebrafish has been recognized as one of the most potential and the most useful model system in biomedical research. Now, there are 4 major applications of zebrafish in biomedical science. The first one is in disease modeling.
Okay.
You are modeling human disease in fish. I'll tell you why and how you can do it. The second aspect is studying functional genomics. So basically, looking at the architecture of a genome, looking at the composition of the genes, looking at how they are placed in the genome, what are the coding regions of the genome, what are the non-coding part, etc., etc. The third one is in pharmacology and toxicology.
Drug testing.
Drug testing. Drug discovery as well. And the fourth one is in the field of, say, looking at environmental aspects of certain important biomolecules that are known to affect, human processes or physiological aspects.
So you mean using zebrafish as an indicator?
Yes, it can be used as a. We in broad terms we call them as model systems, where you can model certain conditions that you would expect to have some effect when you do it in a animal system.
Right.
So at this point of time, as we talk about zebrafish research has undergone tremendous developments, and there are a lot of important discoveries that has happened using this model system.
Right.
Incidentally, zebrafish is a native to India and the adjoining countries.
Right.
You would find them in paddy fields.
Interesting.
You would find them in low-lying areas. They are very sturdy fishes. Another good aspect of zebrafish is that they belong to the largest family of fish called Cypriniformes.
Why is it called cyprini?
Cypriniformes, they belong to the family Cyprinids. Cyprinids constitute a huge family of fishes. There are innumerable number of members in that family.
No, what's in the name of cyprinids?
Cyprinids, it comes from a Greek word.
Okay.
I do not remember the origin of it. But what I can tell you is that the most of the commonly used edible fish in northern part of India, rohu is one of them, which is very popular in Bengal. Katla is another. Zebrafish belongs to the same family. So it has been around for millions of years in the environment. Name zebrafish came from the horizontal stripes that it has on the body. Okay. Yes, it is a fish but it has stripes which are horizontal, unlike the vertical stripes that you find in zebra.
Right.
So that's why it's called
Zebrafish.
zebrafish. And generally, they grow up to 4 to 5 centimeters. That's the maximum size they can grow. And they live up to 4 to 5 years in the wild. And as I already told, they are extremely sturdy. They can withstand a lot of environmental, fluctuation, and they can survive at relatively high temperature as well. So, in India, of course, zebrafish is emerging as an important model system. I'll tell you why it has become popular, or rather why it has gained more popularity in recent years.
Right.
But traditionally, it has been the choice of model for people who are into developmental biology, hardcore developmental biology.
Like
People have been
developmental
using,
biology, what takes place?
looking at cell fate, looking at cell polarity, looking at the way or looking at the processes by which an organ develops.
Is like it's a journey in a
Journey,
timeline
yes,
from an egg,
a single
gametes
fertilized
to-
egg to multicellular individual or multicellular animal, whatever you.
So you mean there is a lot of similarity in the developmental aspect of humans and zebrafish? They like look alike.
Yes.
Can you distinguish a, let's say, 3-week-old human embryo from a 3-week-old zebrafish embryo?
I will put this answer in a different way. Now, as I said, it's a Piscean system. It's a fish. So fundamentally, anatomically, they are different. Zebrafish, the fact that they are not mammalian system, so they don't grow mammary glands. So obviously, there is a difference over there. And they don't have lungs, so they breathe through their gills. So there are fundamental differences. But the advantage of using this model system lies from the structural and functional conservation of genes which are responsible for many fundamental processes in the body.
Like how many percentage of human genome and fish genome are quite the same?
75 to 80%
Oh,
of the genome of
okay.
fish and human beings are similar.
The 15% makes a huge difference from a fish to a human.
Yes.
Okay, that's
But
crazy.
then fish and human beings have diverged in the evolutionary tree about 400 billion years ago.
million years ago.
Billion.
No, Earth formed 5 billion years back.
So million.
Million,
Okay,
million,
years
right.
ago. Million years ago.
Right.
So if that is the conservation, and if these genes have remained conserved over so many years, so that means if something is not correct in fish, is definitely not going to be good for human beings. Zebrafish has a lot of inherent advantages. To answer your question, whether we can compare a 3-week-old human embryo or say a 3-week-old fetus to a zebrafish embryo. Fundamentally, the difference is zebrafish fertilization is external,
Wow, interesting.
unlike human beings. The mating happens outside the body. The sperm is released by the males and the eggs are released by the females. The fertilization happens outside the body and everything is external.
So how do they copulate?
So what we do is we put them in, of course, in natural condition, they are free to move around and, the system could be slightly different. But in lab condition, what we do is we acclimatize them to mate. So zebrafish requires a fixed light and dark cycle. So we generally give them about 14 hours of light and 10 hours of dark. So when we acclimatize them, we put the male and female together the previous night. And when they are put together, the dark cycle gets unset, right? So when in the next day morning, when the light cycle starts, the light or the coming up of the light triggers the
release
chasing
of-
process.
okay.
So basically, the male chases the female,
Interesting.
and they prefer to breed in shallow water. So we have these breeding tanks are made in such a way that there is a contour which allows them to go to little shallow part of the tank where the water level is slightly less, and then they mate there. And once the eggs are released, they are quickly fertilized by the sperm, and eggs being heavier, they fall at the bottom of the tank. So we have a mesh which separates the fish from the eggs. Now, why we need to have the eggs separated? Because they are carnivorous, so they eat their own eggs.
Oh.
That's the
Nature.
law of nature for any
To conserve
smaller-
energy.
Yeah, exactly. Smaller group of animals, and they produce eggs in bulk. One female at a single spawn can produce about 200 to 300 eggs.
Hmm.
So there are a lot of advantages with this. The first advantage is its genetic conservation that I've already explained to you. The second one is its optical transparency. Perhaps this is the most useful and the most beneficial aspect of using this model system.
Right.
Because the fertilization is external, because you don't need to cut open any pregnant animal to get the pups out, unlike in rat or mice,
Right.
you can look at the developmental process without sacrificing any animal.
Most ethical.
All you need is a very good microscope and loads and loads of patience, because you want to see the process of development happening.
Right. Right.
The third aspect is its low cost. If you spend about 100 rupees on setting up a mice or a rat system in a lab, you would spend about 10 rupees for a zebrafish setup. And the best part is the number of embryos, the number of eggs that you get. High fecundity is another important aspect.
Right.
The number of eggs that you get at one spawn from a single female is very high, the huge number, and that gives you the ability to do experiments with a lot of reproducibility in terms of its statistical reproducibility.
The more experiments you make,
The more experience you
data
make,
get.
the more data you take, and the more reproducibility of the data.
Right. Right. And
Can
you can
be
maintain the same strain throughout.
Strain, yes.
Okay.
So these are some of the advantages of using this model system
Over mice.
over mice. And it is small, so you can house them in relatively smaller environment. And the good part is they will not run away. In mice or a rat, there is always a problem of them escaping
Right. There is intelligence problem.
the system, because if the system is not well guarded, if there are some holes in the or some some sort of crevices or something like that, a mice could escape from the cage.
And also psychologically complicated compared
Psychologically
to...
complicated, yes, but then escape of experimental animal is a big headache.
Right.
And it's a big problem as well,
Right.
because you would probably have all kinds of transgenic fish. You would
Right.
have all kinds of genetically modified animals
Getting into the real
getting
world.
into the real world, and they contaminating the natural population. With fish, it doesn't happen because they are always in water. Even if they jump out of the tank,
That die.
they can probably move half a feet or 1 feet. That's it, and then they will
Regret the
not-
decision of jumping.
they will
into
regret
water.
the decision of jumping out of the water. Yes, so that's another good aspect of the using the fish. In India, at this point of time, there are about 15 to 20 major institutes using this system, recognizing its potential. A lot of pharma companies have brought in zebrafish as one of the model systems
is gaining
in
popularity.
their repertoire, in their portfolio. So, in addition to mice and rat, they also house zebrafish, because you can do a whole animal-based experiment with a lot of physiological relevance using a huge number. So what we generally tell you is that zebrafish, because it is a vertebrate model system, which is not yet a mammalian system, we are not saying that it can completely substitute a mammalian system, but it can certainly complement the experiment that you do in a mammalian system. So there is in pharmacology or in drug discovery, we say something called getting rid of unwanted liabilities. So when you do drug discovery, there are thousands of chemicals which are screened, and out of which probably one or two are going to be potential ones which are going to be characterized further. So those 1,000 chemicals that you screen at the beginning, those can be done in a fish model because you are going to spend less money on them. And then you come down to the most useful ones, and then those can be taken to a higher model system. So you save time, you save money, and you save resources. So that is another good advantage of using this model system. Last part is not that ethical considerations are not required. Of course, it is required for fish as well. Till recently, fish model was not part of the animal ethics, purview.
What made them to change that decision?
Because fish, for that matter, any living being has the ability to experience pain. The concept of ethics is primarily into looking at that part because they can't speak for themselves. So any organism for that matter. But then, because the law of nature is such that at certain stages, the way they reproduce is they produce large number and then leave it at the mercy of the nature, and then whichever survives goes on to propagate the population, etc. So with fish model, or say for example fly model, or even C. elegans, which is a worm model, they are produced in bulk. So obviously at a certain stage in their developmental process, it is okay to leave them out of the ethics. But then any experiment that you use, that are going to use zebrafish, particularly the adult ones, are now under ethical purview. It requires an ethical clearance. Till sometimes back it was not, but right now it is included, and I feel it is a good thing that has been done. Because at the end of the day, it's a living being, and despite the advantages, we should be careful about how to not indiscriminately use this model system just for the sake of using it. There should be enough justifications, there should be enough reasons for using this, because at the end of the day, regardless of which stage of development you are using them, ultimately you are sacrificing them for the sake of science, for the cause of science. So obviously we have to be as judicious and as prudent as possible here.
Right. Oh, that's great. I mean, you answered my next question, which was supposed to be why choose zebrafish over mice, which is already there.
Yes.
So you answered it very elaborately.
Right.
Frugality, as far as I recall, variations, ethical, and the cost of running it.
Yes.
Complication, less complicated. Okay. That's why it's getting popular.
But there is a word of caution here. Unlike other model systems, for example, fly, or even for that matter with mice or rat, there is a concept called demand feeder. You can leave them without attending personally. You can leave them, and usually they take care of themselves.
Right.
But that's not the case with fish. With zebrafish, you need to look at them every single day. You need to ensure that, because the system that they are growing is so dynamic.
So this is the unit system.
This is a unique system only because they are growing in water, and the water has to be absolutely suitable for them to live happily.
Like 100% sterilized water?
Not necessarily.
Okay.
But then the water should be good enough for them to get enough oxygen, first of all. It should be less of, it should not be with too much of nutrients, because it leads to high COD and BOD. And then the water should be clean. It should be cleared of the dirt and the feces that are released by the fishes. So every single day we need to take care of them as we would take care of the babies.
Right.
They need personal attention, which sometimes is not required for other model systems. So people could, for example, E. coli can be grown on a slant, and they can be put on a glass tube, and they can be kept in a freezer for months together.
Right.
And whenever you need, you come back and revive it. But this is not the case with fish. So you need personal attention, and
that is the con
you
of
need
it.
to take care. So it needs dedicated attention. But it's a wonderful system to work with, primarily because you can see every process of the development.
Right. So you have worked over many years on this. Have you observed any interesting observation, behavioral wise, of zebrafish?
Every single day has been interesting for me. I started in the year 2006.
Okay. What is your personal journey of discovering zebrafish?
So I did my PhD in Japan, in University of Miyazaki. I went to...
Which year?
I went in the year 2002.
Okay.
I enrolled for my PhD in 2003.
Okay.
I was awarded the MEXT fellowship, which is Government of Japan's fellowship for PhD. I completed my PhD in 2006. Incidentally, my PhD was also on something related to fish, but those are for fin fishes, means the fish that we generally eat.
But
So-
what was your PhD on specifically?
My PhD was basically on looking at taxonomic identity of a group of fishes. They're called ribbon fishes, which are long, slender, silver-colored fish. Worldwide, I wanted to look at the taxonomic identity worldwide using DNA sequence information. So it's called molecular taxonomy. So I looked at the worldwide status, distribution, and to confirm their taxonomic status through their genomic DNA sequence. So morphologically, many people have identified them as different species, but are they really different? Do they have a distinct DNA profile compared to the other species that have been described so far? So those are the things, those are the questions I needed to answer during my PhD. So once my PhD was over, of course, if you enter in an academic research journey, your next step, appropriate step, becomes looking for postdoc experience. So I was looking for postdoc positions in Japan because I wanted to stay there and do a little bit more research. And then I got a call from the Faculty of Medicine from the Frontier Research Centre. They were looking for someone who has some experience with fish.
Japanese Institute?
Yes,
Again.
that was in the same place in Miyazaki. Miyazaki is a prefecture in the Kyushu island of Japan, in the southern part of Japan. So I was looking for a postdoc position, and they were looking for someone with an experience in handling fish.
Right.
So it kind of
So
matched.
it wasn't specifically zebrafish.
It was
Zebrafish, okay.
Just that in my PhD I didn't do zebrafish. My experience, or I got introduced to zebrafish
In your
only
postdoc.
when I entered the postdoc. So I went to that lab, and then they were already doing fish, zebrafish based research. So that's how I got introduced to the system. And then I quickly realized that this is the model system that
Right.
I'm going to work with for the rest of my life, hopefully.
Oh, there you made your mind.
There I made my mind, because it was quite fascinating to see how organ systems develop without
Involving humans.
without involving humans, and without getting into too much of complications. Simple instrument will help you identify or will give you an idea to understand the intricacies of life.
Right.
So that was quite interesting, and that was quite exciting as well, to see how the eye develops, how the digestive system develops, how the brain develops cellularly, and how the entire process of the development from the first cell to 2 cell to 4 cell to 8 cell to 16 cell, and then how the entire body is taking shape. So these things were unheard of. We would have never imagined that we could see them in real time. So that was something quite exciting, and that's how I made up my mind that I would work with this system, and that's how I got introduced and entered into the world of zebrafish.
When did you come back to India? And did you get a zebrafish along with you as well?
No. Unfortunately, I'm not permitted to bring fish.
Okay.
Of course, there are procedures to be followed. There are ways to do it, but then since scanning live animals is a big challenge, so you can't do that. Even if you follow the legal procedures, they will not survive because it's a very complicated thing. Not to forget the temperature at which the aeroplane flies. It's impossible for a life to to remain alive. Anyway, so I came back in 2014.
What changed your mind?
Couple of things. One was the most important or the most compelling reason was perhaps it was more personal. My mom was alone and she was finding it difficult. So perhaps that was the most compelling reason. And I being the only son, I felt that I have some responsibilities. And since I spent almost 10 years in Japan and then about 2.5 years in France. From Japan, I moved to France. And of course, it was almost quite certain that I would settle down in France. There also I went because there was an opportunity to work with Zebrafish. The concept was slightly different, but then it was still the same model system. So that kind of attracted me and I went there and I was quite comfortable. But then something happened at personal level and I lost my dad and then mom started feeling alone and that was the most compelling reason. Second thing was to perhaps the urge of coming back and to set up your own lab, to set up your own facility and to
have your
impart knowledge and skill to people who can benefit from this system and who can go on to do good research. So those were also there in the back of my mind and so all these helped me decide to come back and set up a lab. And all my life after my PhD, I did perform the role of a postdoc, and then I was a junior mentor for some PhDs and etc. But then it was always like I was working for someone, and once you become a postdoc, you develop those skills of being independent, and you always want to set up your own area of research, and you also want to
your own lab.
You want to make your, of course, you set up your lab, and you also want to build your team with people. And so that was also one of the things that I could have done if I had stayed back. But then the opportunity that came by was good enough for me to take it up, and that led to the decision of coming back to India.
What was the status back then in India in terms of zebrafish research? Like, was it really popular back then, or?
Yes, the answer would be both yes and no. As I said, about 20 to 25 premier institutes in the country right now have the zebrafish facility. And these are all government institutions, right from CCMBs to ICERs to IISc to NCBS to IGIB, etc. These are all premier research intensive institutes that set this up. But in the private universities, it was still growing. It wasn't so popular at that time. Certainly, when I came back to India in 2014, obviously things were not as it is right now. We didn't have too many labs, particularly in the private universities, who would have even heard of zebrafish. Forget about having one. And the same thing happened in this university as well. When I came back and when I expressed my desire to set up a facility, I mean, people are not very sure whether the investment would be right, and because they didn't know about it. But then because of my past credentials and because of the past research that I did within the same system, they got convinced and they helped me set it up in a very primitive way. I must acknowledge the help of my classmates, Ronald and Ashwin, who are running a very successful aquarium business in outskirts of Mangalore in Vamanjur.
Nice.
They're running a very successful business. Their company is called Aquatic Biosystems. So they helped me set up a zebrafish rearing unit with the design that I gave, and then they helped me build it. So I started with that, and in fact my first fish came from their lab, from their farm. So as I said, zebrafish, you find them in the wild. It's actually wild fish. So they collect these fishes from various rivers and other places.
Right.
And then I started with that, and I had to set up the system of how to breed them, how to mate them, how to raise the embryos, because it's a very critical step. It's not just working with adults. You have to work with 1 day old to 2 years old, 3 years old fish. So you have to have the entire process streamlined, and you have to have a system where potentially you can grow even a single embryo that is fertilized into a full-fledged adult. So I had to set that up. I had to make the facilities functional. And once it got functional, I got in touch with the scientists who are already working with fish in various other institutes. They helped me get some lab-bred lines and some specific lines related to the research that I pursue. Yeah.
So you said you spent 10 years in Japan. So do you know how to say zebrafish in Japanese?
In Japanese, it's zebrafish only. It's the same name. Yeah.
Right. So what do you call it locally? If you say Kannada or Tulu or any other languages?
I don't know.
No idea.
Because it's there must be some local name, but since it's a weed fish, it's not eaten. So perhaps people are not very keen on giving it a name.
Right.
Yeah. Yes, I'll just answer the other question. Fish in Japanese is sakana.
Okay.
Okay. Yeah.
Zebra sakana.
Okay. Yeah.
So what do you call zebra in Japanese?
Zebra. Zebra is zebra.
Okay.
I think it's not a native of Japan.
So what do you call black and white in Japanese?
Shiro is white. Kuro is black.
Shirokuro sakana.
Shirokuro sakana? Oh, okay. Could be, yeah.
Just kidding. Okay. Right. So, yeah, back to the interesting observation on their behavior.
Behavior, yes. Zebrafish, they love to chase and eat. So that is one thing. So they love live feed. So when you give a feed, it has to be a moving feed. So that's their nature. They chase and eat. So they're quite aggressive. The males are, as probably is the case for any species,
right.
The males are more aggressive than the females. And then sometimes if they are stored in, or rather if they are reared in big tanks where they are big in number, in terms of say 50, 60 fishes put together, sometimes they exhibit the shoal movement. Shoal means they move in a group. So if you move your hand from the outside the tank, they would move along with the movement of
wow.
hand.
Okay.
So that is nice to see, as if they are following your orders.
It's like magic.
So
Disney
you
World.
want them to move this way, they will move this side, and then from right to left, they could turn back and swim again. Yeah. So that is a nice swimming trend. And for any fish, for that matter, if they are in a very comfortable condition, they'll be swimming in the middle of the water, not at the bottom, not at the top. If they're at the top, that means they are not happy. They are grasping for air or things like that. So perhaps they will come to the top only when there is a feed or something. Otherwise, they'll be in the middle of the.
It's a comfort zone.
Yeah, that's their comfort zone. Yeah. And then there is something like what we generally do when you do experiment that's called escape response.
Okay.
So what we do is, so they have this. Let me go back to the time when they are 1 day old.
Okay.
So we call this DPF, days post fertilization. And when we express this in terms of hours, we call it hours post fertilization. So HPF or DPF. So when we are looking at developmental changes happening at an hourly basis, we call it at HPF. So for example, we have a board over here. You see this board. This was done by our students. Her name is Carolyn. So she hand drawn it.
Nice.
And the ones that you see behind you, these are all the stages of zebrafish you see the first one on top is 24 hours.
Right.
And then the second one is 48 hours. And the last one is day 5 or 5th day. So it is about 120 HPF.
Like a larval stage, is it?
Larval stages. How the organs are coming up. And if you see, there are 3 visuals, 3 ways by which it can be picked. So one is the dorsal view, when you're seeing the fish from top. Middle one is the lateral view, when you're seeing the fish from the side. And the last one is the ventral view, when you're seeing the fish from the belly.
Bottom.
So upside down, you're seeing from the belly. So if you see the middle one at day 1 and the middle one at day 2 and the middle one at day 5, you see so much of difference over there?
Yes.
In terms of the organs coming up, etc. So when they are born within a protective covering. It's called a chorion. It's a membrane which is a protective covering. And then by the time they are 2 days old or 3 days old, they break this cover open and come out.
Right.
So if they are unable to do it, it is considered as a developmental delay. And it is an important milestone in the development. So they should be able to do it on their own. So by day 3, all the fishes should be out of that cover. It's like hatching out.
Right.
So that stage is called hatching stage. And that coincides with their ability to look for food. Because by 3 days, the mouth development is complete. So once the mouth is developed, they start looking for food.
Oh, immediately.
Immediately.
Nice.
Otherwise, even if the food is available, they will eat. If it is not, they will not die because the yolk will still sustain them for about 4 to 5 days. By then, the digestive system starts developing, etc. So when they are within the chorion, in day 2, they start showing the twitching movement, as you would expect a fetus in an amniotic fluid,
right,
moving around, something similar,
kicking.
So they will have this twitching movement inside the chorion. So this is very nice to see, as how quickly they move.
That means the eggs are transparent.
And the eggs are transparent, yes. And you will see a fully grown fish all curled up inside that protective bubble.
Right.
Just like a human baby. It's all curled up. And once they come out, they are all straight. Because this is a fully developed embryo. Nice. But when they're inside this, they are all curled up. So when they twitch inside, it's a very nice thing to see. And once they come out of it, then there is something called, if you touch the tip of the embryo, tip of the tail, they have this escape response. They move out of the focus very quickly. So that escape response is a spontaneous movement. And that is a natural movement. So we make use of this behavior as an observation if we are to be looking for a neurological defect or a muscular defect. So
it's like Alzheimer's version of the fish.
Something like that. So when they are not able to move, so when they are not able to show that escape response, there will be 2 reasons. One could be muscular defect or it could be the nervous defect.
Right.
So this is a very nice thing that you can do very easily. So when we look at the toxicity of zebrafish, so basically we look at 4 developmental stages in the embryonic stage.
When you study toxicology of zebrafish.
So there are called these toxicity assays that you do. And there are about 28 to 30 endpoints, what we generally check. So these are endpoints, means whether they are lethal or sublethal.
Okay.
I'll give you one example. Heartbeat at 48 hours after they are born. If the heartbeat is normal. If the heartbeat is slow or fast, then it is a sublethal effect. If there is no heartbeat, it's a lethal.
Okay.
So like that, we check about 28 to 30 such endpoints.
What are the 5?
So in day 1, once we look at whether the brain development is complete and whether, as I told you, if you look at the development of zebrafish, It is everything starts with one cell, right? Fertilized
Right.
cell, and then when the process is going on, the entire how do you say, the animal pole engulfs the egg, because half of the egg would be the yolk. Only one part will become the animal pole, remaining part will be the vegetal pole. So the entire thing gets covered and it takes the shape of the fish. So if you look at them at 18 hours or so after they are fertilized, you would slowly start seeing the somites. Somites are basically the muscles, the muscle fragments that we see.
Right.
And then the notochord will come up. Notochord is the
Vertebrate.
spinal cord. Right. Yeah. So at day 1, we see whether the tail is still attached to the yolk or not. The tail should be detached. That is one of the development points. Then at day 2, at day 1, we also look at whether the brain development is complete, where all the 3 parts of the brain, say midbrain, forebrain, hindbrain, whether they are completely formed,
Right.
whether the boundaries are clearly specified, etc. In day 2, we look at the cardiotoxicity, whether the heart looping is okay, heartbeat is fine, whether the circulation is good, blood cells, because they're transparent, you can actually see the blood cells.
Right.
You can even count.
Nice. What is the color of blood?
Red,
Red.
because they carry hemoglobin. So it's the same color. And then
Day 3.
day 3, neurotoxicity. And
Brain
look-
development.
by development. So that is a gross visual observation, where we look at this escape response and their movement, etc., and this twitching movement, etc. And day 5, we look at hepatotoxicity, which is the liver toxicity. So these are standard assays that we do.
How do you guys access liver toxicity? Do you guys feed them with alcohol or something?
No, we don't need to, because as I said, they are transparent. They remain transparent for about a month.
Okay.
So you can see the entire gut development.
Oh, including liver.
Liver is the largest organ there. So you can see very clearly. And if there is any toxic effect on the liver would look cloudy and it will look dark in color, etc. So
Right.
otherwise, it will be transparent.
So
With
it's just
experience-
morphological observations.
It is a morphological. With experience, you'll come to know which part of the body is the liver.
Right.
Initially, you will not be able to find it so easily, but then over time, you will be able to see that. Yeah.
Interesting.
So these are some of the assays that we do when we are using the embryos. But when we use the adults, there are organ-specific assays. There are so many other things that we do.
Right. It can go to cellular level as well. there.
Absolutely.
Right. So my next question is based on, you said it can be used in 4 major ways.
Yes.
One is disease modeling and functional genomics and drug discovery and environmental aspect.
Correct.
I want to cover one by one in a great detail.
Okay.
So let's start with the disease modeling.
Right.
So how can you model a human disease in zebrafish?
Okay.
And first, before that, I think what diseases can be studied and has been studied using zebrafish.
To answer your question, there is hardly any disease that has not been modeled in fish yet. Almost every disease has been modeled.
Even Parkinson's.
Even Parkinson's, even cocaine addiction
Wow.
has been modeled in fish.
What is the response? Even they're addicted to cocaine?
Just few days back, I don't know if you have read this, sharks of the coast of Brazil were found to be positive for high doses of cocaine.
Oh, expected in Brazil, right?
Have you read this? Sharks.
No.
They are already aggressive. With cocaine, the expectation is that they are going to be even more aggressive.
We should make a next movie on this.
Anyway, so what I'm saying is, fish has been used even to model insomnia. There's a general perception, people feel that fish do not sleep. They do sleep, it's just that they do not have eyelids.
Right.
So you do not realize that they're sleeping, but fish do sleep. So insomnia is also been modeled in fish.
Wow.
So practically,
Every disease has
every
been modeled.
disease, genetic condition, particularly the genetic diseases, are very well characterized in fish. You can also study infectious diseases, effect of Corona also, yes.
Wow.
Incidentally, I have a project right now funded by Indian Council of Medical Research, where we are looking at genomics of patients to try and identify if there is any specific variation in the genome that correlate with the severity of the infection in them. Because in corona, we had severe people, we had mild people, and we had moderate people, right? I mean, the degree of severity was...
And carriers.
different kinds. So this particular project is about identifying this and then modeling in fish. So we are developing mutants in fish for 2 genes that are responsible for attachment of the SARS-CoV-2 virus to the cell.
Respiratory cells.
Any cell for that
cells,
matter.
okay.
Where they attach, there are 2 genes. One is ACE2, which is responsible for the viral binding and entry. And TMPRSS2 is another gene which helps in the transport of the virus inside the cell.
Right.
So we have, the project demands that we develop mutant for these 2 genes in fish, which are supposed to be for human beings. But we are going to study, we mutate them in fish, and then we inject them with a protein from SARS-CoV virus, because we can't handle the virus. So we are injecting a protein which is supposed to be important in the pathogenicity of the virus. And see whether if you are injecting that protein into that mutant, what kind of immune response you are seeing. Because people died in corona or COVID because of the hyperactive immune response.
Right.
They didn't die because of the viral infection. Overstimulation of the immune system that led to something called cytokine storm. And there is a pathway which is known as RAAS pathway, which involves these genes that are also important in viral attachment and entry.
Right.
And these genes end up in a pathway that has major complications or consequences in kidney, in liver, etc., the major organs. So people, the hyperactive immune response is what we are going to study in fish, which is going to be related to an infection that is supposed to be in human beings.
Right.
Another important example of an infectious disease I could give you is studying tuberculosis in fish. So the main bacteria that causes tuberculosis in human beings is called Mycobacterium tuberculosis. But it's, it's a brother or sister, I could say.
Variant.
It's similar species, not the same species, but something similar in the same family. It's called Mycobacterium marinum, which is a natural infectious agent for fish. Fish is a natural host for this. So instead of studying or instead of handling TB bacteria, you can handle marinum. Marinum will not infect human beings.
Equivalent in fish.
Equivalent. And the kind of granuloma that is formed when a person is affected by TB is the same kind that you would see in the fish when the fish is infected with Mycobacterium marinum.
Interesting.
So we use it as a surrogate marker for studying Mycobacterium infection of human beings. So these are 2 examples of infectious diseases. There are plenty more people have studied. Coming to genetic diseases, there are hardly any genetic disease that has not been modeled. Right from your diabetes to your Parkinson's to Alzheimer's to all kinds of obesity, non-alcoholic fatty liver disease, alcoholic fatty liver disease, rare genetic conditions affecting the stem cells, affecting the hematopoietic stem cells, affecting the blood cells, etc., etc. So many things have been.
What about chromosomal disorders like?
Those were also.
What is the observation there? Is quite interesting.
It's quite interesting because, see, now when we use a model system, one thing we need to know is, particularly when you're working on genetic models, how amenable the model is to genetic manipulation. If genetic manipulation tools are going to be easy in a particular model system, that model system becomes a preferred one. So there are plenty of genetic tools which are available, and the latest and the most effective one, which is known as CRISPR, is being very effective in fish. And nowadays, people have started using CRISPR for almost all kinds of genetic manipulation. Earlier, people are using different... So there, in broad category, there is like something called forward genetics. You first randomly mutate the DNA, and then you identify through the phenotype, and then you go back and find out what is the cause.
Right.
But there is something called reverse genetics. When you target a particular gene through a specific technique,
Okay.
like molecular scissors, we call it, but we target a specific gene. We want to inactivate that gene only. So there are many techniques like this. There is zinc finger nucleases are there, and CRISPR is another, morpholino is another,
Right.
then TALENs are there,
Right.
Tilling method is there. There are so many of them. So these things, these techniques help you identify a very specific gene, and you look at the consequences of that gene deletion.
Right.
So these techniques are quite amenable in fish.
That's how we study genetic disorders in fish.
Exactly. And then the third thing comes about your chronic conditions, your toxic effects, adulterants, preservatives, colorants, so many of them which are supposed to be carcinogenic.
Right.
So these things can also be studied very
This is
easily.
important. Like we don't know
Histologically-
what are the carcinogens out there.
Exactly.
Maybe
So-
in our perfumes, it could be our
Could be,
food.
could be, yes. One good thing about the fish is the histological changes that you see in a cancer development, the pathological changes at the cellular level. If you see a zebrafish tissue and if you take a human tissue, they are identical. So the process at which they are changing and the way they are changing is identical. And the genetics of it is also conserved. So gene which is supposed to be suppressing tumor growth in human, the same gene has the same function in fish.
Wow.
So if you remove that gene from fish, that fish will end up with cancer.
Right.
So
Like they have the same genetic
same
code.
genetic code.
Wow.
Yes,
Interesting.
yes. So there is a gene called P53.
Okay.
It's the most important tumor suppressor gene. So if you remove P53 in a cell, human cell, or in a human being, there's a very good chance that the person will end up with some kind of cancer
Right.
at some stage. Same thing happens in fish.
That's how you model cancer studies in the zebrafish.
Yes. So there are many other things that you can do, not only just looking at the growth of the tumor, because in cancer, the primary tumor is not the only problem. Because if the primary tumor were to be the only problem, people would get rid of it and get an organ transplant.
Right. It's
But
the
that's not
spread
easy.
of the tumor.
It is the spread of the tumor which makes it a deadly disease. So there is no way people would know whether the tumor has started moving in the body. And that process is called metastasis.
Right.
So if you develop a drug which can prevent metastasis, that could be an anti-cancer drug. If you develop a drug that can prevent the formation of blood vessels, new blood vessels, because when cancer moves to a different part of the body, when the tumor is growing in a different
they need a
site,
supply of oxygen.
it needs blood vessels for oxygen, for nutrients. So if you can stop the formation of blood vessels, that is also a good anti-cancer strategy.
Right.
So these strategies can be very well employed in fish.
Right.
So we have some transgenic fish where we have a green fluorescence protein which is obtained from jellyfish, and by transgenic approach we can get it integrated in the zebrafish genome
Make them glow.
and make them glow only in the blood vessels.
Wow. Interesting.
Right? Because we are interested in looking only at the blood vessels.
Do you guys have such samples?
Yes, we have a sample.
Wow.
We'll
go through it later.
We have such animals where
Nice.
the blood vessels glow as a green fluorescence protein.
What does that indicate? Whether if it glows and if it doesn't glow?
If it doesn't glow, that transgenesis is not happening.
Isn't happening. Okay.
If it is glowing, that means, so how you make it glow is you make this protein express in that particular organ through a gene which is meant to be expressing in that organ. So you know what are the driving factors of that gene. So you clone that part of the gene, put it in front of GFP by genetic stitching, and then you inject into the fish, and then once you inject it, you expect germline transgenesis to happen. So when you inject in a cell, foreign DNA, the DNA is, as the cells are dividing, DNA also gets distributed.
Right.
So by chance, if it gets into the germ cells, the analogs of the germ cells, which are going to be the germ cells in future,
Right.
and if it has gone in there, then there is a germline transgenesis that is going to happen in the next generation.
Right. Even the embryos will grow.
Exactly. So to check whether transgenesis has happened or not, we inject into a fish, allow the fish to grow at least 3 months because they have to be adult. Once they have grown for 3 months, then we mate them with a normal fish. If half of these embryos glow, that means the primary injected fish
Right.
has undergone germline transgenesis.
Okay. Even you can study the germline distribution, whether this gene is dominant or recessive.
Yes.
That way. Wow,
Everything,
interesting. And my question here is,
So-
when you study cancer, is even the metastasis, the spread of cancer cells, is that also similar to human cells, like how it spreads in humans?
There's a technique called xenotransplantation.
Okay, external organ transplantation.
Yes. So what we can, zebrafish is a good model for that.
Wow.
For two reasons, because at early stages the pigmentation is very minimal, so you don't have any interference with the pigments. And the second part, as I said, they are transparent, so you can see the movement of the cells. So people, what they do is, you can take human cancer cells, which are highly metastatic, and you can inject into the zebrafish, and you can see the movement of these cells. Suppose you are trying to discover a drug which is supposed to stop this spread. You can have two systems, one without the drug, the other with the drug. So if the drug is really effective, next day you would see the cells are still localized in that place.
Right.
But in the other one, they would have moved to different parts.
Is that does that cancer cell movement as pattern, or is it just random? They randomly settle somewhere.
Depends on the kind of the cells,
Right.
what you are injecting in. But invariably they will move through the blood vessels. So obviously wherever the blood vessels, whichever direction
Right.
the blood flow is, cells will move that way.
Okay, so you can see that clearly in zebrafish.
Very clearly, yes.
Yes. So how do you study obesity or diabetes in zebrafish?
There are the two ways of doing it. One is you can induce obesity by diet, or you can have a genetically modified animal which is prone to diabetes.
Right.
So generally, depending on the need. So when you say diet induced, you give them a high fat, high carbohydrate diet. So within maybe 1.5 to 2 years, they will end up as obese
Right.
fish. In fact, zebrafish is a very good model for studying atherosclerosis. Atherosclerosis is formation of cholesterol plaques in your blood
Vessels.
So
Why is it a good model?
because you can very easily develop the system, and you can actually monitor the effect of the drug, anti-cholesterol drug, at a real time in this fish, using the embryos, because they are transparent.
Right.
So you can see the movement of the blood cells, and you can see the architecture of the blood vessels. So if you use a confocal microscope, which can give you a very high resolution of your blood vessels, you can see and of course, if you add a mimic of a cholesterol which has this ability to glow, so you will see where it is getting deposited.
Right.
And you can also, as a proof of principle, if you do an anti-cholesterol drug, you will see that that deposits are not there in the fish which is treated with the drug, but it is there in the fish which is not treated with the drug. And atherosclerotic plaques are the main reasons for
Heart attacks.
the infarction that we talk about.
Right.
So blockage of the arteries is what leads to the heart attacks, right?
Right.
So that can be very easily studied in fish. So like that, there are innumerable examples of how a fish model can actually score better over mice and rat model.
And how would you?
There are some diseases where rat model or mice model are not suitable at all.
Like?
For example, there is a very rare condition called diamond blackfin anemia. Diamond blackfin are the doctors who discovered this condition. Now the children have the most common clinical manifestation is anemia. So they are highly anemic. Now children are heterozygous, means when you sequence their DNA, you will find that only one copy is defective, the other copy is intact. So if you want to simulate the same thing in a mice or a rat model, the mice or the rat model will not show a phenotype because there is a compensation that is happening. On the other hand, if you remove both the copies, the homozygous ones,
Right.
they will not be even born because they'll be dead, embryonic fetal. But in zebrafish, because they are external, the embryos, the homozygous ones also die, but they die after a period of 7 or 8 days.
Mm-hmm.
So you can continue to watch them for 7 or 8 days.
The whole process.
Whole process, which you cannot do it in a mice or a rat. Because if the pups are removed, they will be stillbirth.
Right.
Because the homozygous ones will not be born. They will be
Right.
a stillbirth.
There's a lifespan disadvantage also in the
Lifespan, but the most important part is homozygous ones, which are embryonic lethal, will remain lethal even before they are born.
right.
But in fish, they are also lethal, but you get about 7 to 8 days.
Mm-hmm.
7 to 8 days is a big window to study their
Right,
physiological
right,
consequences.
right.
Yeah. So these are in some cases they are actually advantageous
Right.
over mammalian system. Yeah.
So how do you study neurological disorders like aging, Alzheimer's, Parkinson's?
There are so many ways by which you can look at the behavior of the fish. You can have a lot of systems where they have even zebrafish is also believed to have numerical memory. They
What do you mean
can
by that?
numerical memory means they can distinguish between
Space and time?
space and not just that numerical. If you are keeping 5 chunks of feed
Mm-hmm.
in one tray, and if you are putting 3 chunks of feed, they will go to the feed which
Oh, nice.
5 chunks of feed. So that has also been studied. So there are a lot of behavioral essays that has been kind of designed, and so many of them have at the behavioral level you can do it,
Right.
and of course you can look at the level of the hormones which are involved, which are deregulated. You can even have the sectioning of the brain done, etc., etc. All these are routinely done. People are working, and of course you can look at the structure and the function of the neurons
Right.
by looking at these. There's a big team in NCBS in Bangalore, Dr. Vatsala. Her team is into neurobiology. She is working on looking at these neural interactions and all of
Right,
that. So
right.
I think she's the only one in the country right now working on this.
So you mean you can also induce coma in...
You can, exactly. You can do that, yes. In fact, routinely we anesthetize them as we do it in a patient undergoing an operation. In fact, there's a technique where, by the way, I didn't tell you, zebrafish has this unique property of organ regeneration.
Oh, okay.
They can
like every organ, or there are some
almost
specific
all the organs. They can regenerate their heart,
Okay.
they can regenerate their kidney, and then fins, of course.
Right.
So one of the techniques that we do is we cut the tip of the tail
Mm-hmm.
to identify their genetic makeup. So when you cut the tail off, within 7 to 10 days, the tail comes back.
Right.
So it is a good model to study wound healing,
Wow.
also to study regeneration.
Right.
So when we cut the tail, we anesthetize them. Otherwise, you'll not be able to put them.
Right. Right.
You will end up murdering them.
Right.
So what we do is similar to as it is done for patient. So we put them in a anesthetic agent. We put them to sleep. Then put it on a tray, cut the tail, and put them back into the tank with good water, and then we aerate them quickly for few seconds, and then they revive.
How would you study heart regeneration? Like you have to get inside the organ and then cut it and then stitch it back?
Yes. So there is a team in Agarkar Research Institute in Pune.
Okay.
Dr. Chinmay. He is a pioneer in heart regeneration studies. So right now, most of the people are using cryogenic injury as an injury model, which is not exactly similar to infarction, myocardial infarction.
Like cold
It is a cold, So you induce a cold shock, so part of the heart gets injured, and
Good
then
idea.
they look at how it regenerates. And of course, for that you need to cut open the fish. Yes, one of the simplest examples of um such studies came up way back in, I think, maybe 15, 20 years back. It was published in the journal called Nature,
Okay.
where people, so the experiment was very simple. They took some zebrafish, they cut a part of the heart, stitched it up, and left it in the tank.
Okay.
After a few days, they caught the same fish, opened the heart, and they saw the heart is regenerated again. So
That was the first
that was one of the
discovery.
pioneering works where people actually realized that they can regenerate.
Right. I mean, how studying heart regeneration can help study human diseases?
Oh, plenty of applications. If we know how heart regenerates, and if we know what are the proteins, what are the pathways involved, someone getting affected by a myocardial infarction, the cardiovascular diseases, which is a big burden in a country like ours,
Right.
I think we can have so much of headway into it. Yeah.
We can invent new way to
New way
strengthen
to
our
even-
heart.
Yes, we can also find out different ways of actually predicting the propensity of developing such conditions.
Right.
In fact, just to add here, we have a collaborator from Russia who is an expert in imaging, microvesicle or microcapillary imaging. So we are actually working on a technique which is a non-invasive technique through which we can actually predict the risk of heart attack for patients by just imaging a microcapillary in their arm.
Like change in the pattern of capillaries
In the arm.
Interesting.
So, and to do that, the model that you're using is a zebrafish. So you want to have it, the proof of principle done in fish, and then try to see how it goes with the patients. So this is something that we have initiated.
Right.
In fact, his name is Alexander. He is scheduled to visit India in the month of November. We will have a workshop on non-invasive methods to detect cardiovascular conditions.
I'm pretty curious more on, you said substance abuse can be studied in zebrafish.
Yes, yes.
Like cocaine addiction, alcohol addiction.
Yes.
What about it? Tell me more about it. Like how it can exactly be studied and what can we know more? I mean, what new knowledge we can get from studying zebrafish models?
Basically, for any model system, I think there are three important things that we look into. One is whether the disease that we are trying to model in the fish, whether it is recapitulating the phenotypes that you see in patients. Second, whether there is the pathogenicity or the pathways that are involved, whether the same pathways are getting involved. And the third thing is whether the treatment strategies or therapeutic interventions that people look into can, in principle, work in the model system or not.
Is it effective or not?
Effective or not. So, what new you can study is you can think of developing new approaches. For example, in rare genetic disorders where there is no definitive cure, people depend on steroids. And steroids has their own
Side
set of
effects.
problems. So, if you want to identify new therapeutic strategies, you could potentially identify something in a model system like this. You can discover something which can reduce the dependence on steroids for the patients.
Right.
Something else could be given which could work in a synergistic manner
Right.
with the steroid. So you can reduce the dose of the steroid. In terms, you can look at having less side effects on the patient. So something like that can be done, and depending on, I mean, the way the field has advanced, I think there is hardly anything that I think that we do not know about.
Okay.
And more and more techniques are coming up, and people have done cardinal work. People have done seminal work on this model system.
Right. These are our living knowledge generators, you can say. I mean, we discover new knowledge.
New knowledge,
A lot
yes,
of
exactly.
knowledge.
A lot of new knowledge has come forward. I can't speak for the developments that are happening in other field, primarily because as a researcher, it is impossible to keep track of
Right, everything.
But then I can speak for the developments that are happening in the area that I work in.
Right.
And people have. So one of the advantages of Zebrafish is its amenability to a technique called high throughput screen.
What is that?
It's called HTS.
Okay.
Short form is HTS. High throughput screen means at one go, you are potentially screening thousands of compounds or chemical libraries. People generally use cell lines, which are isolated cells in a 2D environment.
Right.
And that is used for screening. Whatever gives you a best hit, you take them up and then you characterize them further.
It's like time intensive also, right?
Time intensive as well. But then, because you're using a cell, just an isolated cell, what you see in a cell need not work in a whole animal approach, right?
Right.
Because when you're looking at the whole animal, there are many other factors which are involved. For example, if you talk about cancer is not just an event that is happening in a cell. Cancer means a crosstalk between cell and its extracellular matrix. There's a lot of crosstalk between the environment that the cell is in.
Complexities a lot.
So when you look at an anti-cancer drug in a cell, just a cell, the effect that you see there need not replicate same way when you are using it in a animal or potentially on a patient. So what Zebrafish helps is, because they are amenable to this HTS technique, so basically they can be utilized instead of cell lines. Zebrafish can be used as a whole animal model, which will have two things. One is the physiological relevance of the whole animal, and also the reproducibility of the assays that you are doing. Because when you do HTS, you generally use some plates. These are called microtiter plates. These plates come in different sizes. You can have the surface area remains same of the plate. The surface area is same, but individual wells surface area changes depending on what kind of plate you are using. If you have a 12 well plate, the entire plate will have 12 wells. If you want to use a slightly smaller well, you can go for 24 well plates. So the same surface area, but instead of 12 wells, you have 24 wells. Now, the surface area of individual well has come down. Similarly, from 24, you can go up to 48. From 48, you can go up to 96. From 96, you can go up to 384 wells. So in a single plate, you have 384 small wells, where potentially you can put 1 or 2 3 to 4 days old zebrafish. They can survive in just 50 microliter of water. You do not need to add any feed. You do not have to change the water. And they can survive like this for 2 to 3 days.
That's how screening is done, high intensity screening.
So that possibility is unthinkable for a rat or mice.
Right.
You cannot do this. So that is another big advantage.
And it's a statistical advantage as well.
Statistical advantage. So you can screen a huge library, a big library of compounds, several thousand compounds within no time.
And also minimizing errors, because there's a huge samples.
And you are maintaining the physiological relevance because you are using a whole animal.
Right.
You're just not using an isolated cell.
Right.
So that is another advantage.
Okay, what is the most interesting project you have heard of using zebrafish? I can tell you mine. When I looked at the internet, it said in January 2007, Chinese researchers from Fudan University genetically modified zebrafish to detect estrogen pollution in lakes and rivers, which is linked to male infertility. The researchers cloned estrogen-sensitive genes and injected them into the fertile eggs of zebrafish. The modified fish turned green if placed into water that was polluted by estrogen. It was like a real-time indicator.
It's a biosensor, you could say.
Right, biosensor. Yeah. It's interesting.
It's interesting. But only thing is, anything that works on the same endocrine metabolism pathway essentially will end up glowing. So the fish, per se, it can detect estrogen.
Right.
But probably similar hormones will also be detected by the fish. So one good thing about these biosensors is that they can help you identify at a real time, but then there is also the lack of specificity.
Right.
So that is one hitch that we have.
Mm-hmm.
For me, an interesting thing would be, which incidentally has come out from one of the PhDs that worked with me.
Oh, from Nitte University.
From
also.
here.
Okay.
She is right now working as a postdoc in NCBS, Bangalore.
Okay.
She's incidentally working with Vatsala,
Mm-hmm.
who is an expert in neuroscience research in the country. So she did a PhD with me and she developed a mutant which does not show any phenotype except that it turns out to be males. Only males.
Can you repeat, sir?
So we developed a mutant fish.
Okay.
So the fish has one gene which is defective, and both the copies of that gene is defective in that fish.
Okay.
Because we get two copies, right?
Right,
One
heterozygous.
one from the mother and one from the father. So that fish, if both the copies are defective, we call it homozygous mutant.
Right.
If one is defective, we call it
Heterozygous.
heterozygous. The fish which is homozygous, it has no phenotype. It is absolutely similar in all other aspects compared to the heterozygous and the wild type.
Okay.
Wild type means when everything is
Right,
normal,
right.
intact.
How come it is?
But it does not become female. It becomes only males.
Hmm.
So that was a very surprising observation for us, because we expected the fish to not survive till adulthood. But we found that it not only it survived till adulthood, it was a fertile male. It was able to reproduce.
Wow.
But it turned out to be only males.
Male.
Males,
Okay.
only males. So generally, fish is born as female.
Okay.
So sexual differentiation happens during the process of development.
Is it a temperature sensitive?
No, it is a normal process of development.
Mm-hmm.
At some stage, the eggs are bipolar, so it will decide which pathway it will take, whether it will become the testis or the ovary, right?
It's
So-
just pure random, is it?
Yeah.
That
Depending
process.
on which transcription
Okay.
factors express at what time. So there are some pathways. We'll not get into the details.
Right.
But fish, generally they are born females. They divide at some stage, and then half of them become male, another half become female.
Right.
So that's the normal process.
Okay, that's
But
how
in this
differentiation-
mutant, 100% of them become males.
Mm-hmm.
They didn't become females.
So that
So why it was interesting for me? Because when we talk about aquaculture, fish, what we grow in the cultured ponds, people go for monosex culture. Monosex means you grow only one type of fish, either male or female. The idea is to avoid them into mating, thereby not wasting energy, and thereby diverting that energy into growth.
Right. Not
Okay.
on fat storage. or
Yes.
something.
So that energy is stored in some other way, and invariably that will reflect on a growth.
Right.
So in monosex culture, people use hormones for the sex reversal.
Mm-hmm.
But in this case, we found a way by which, if we modify the gene of the same fish, we are not introducing something else from outside.
Just
We're just
one gene.
modifying just one gene.
Wow.
We're just modifying, and we can convert a whole set into a male
Monoculture.
population,
Mm-hmm.
a monosex culture. So
Right.
this can have a wide implication in the
Aquaculture.
aquaculture setup.
So you mean it can be also applicable to other fishes as well?
It can be, potentially.
Oh, nice.
And because we looked at cyprinids, I mean zebrafish belongs to the same family, perhaps it would be applicable for rohu, katla and other fishes.
Wow.
So this is something quite interesting for me. Which
So
I
have you
found-
guys worked on the commercializing? Have you guys
not yet,
taken
but this
further?
technique is going to be tried. Just to add here, Government of Karnataka, the Department of ITBT has sanctioned a big project to us it's a center of excellence on aquamarine innovation. It's a 3 years project which comes with a big funding, and there are 4 verticals in it. One is research, the other one is innovation, the third one is capacity building, and the fourth one is outreach. And we need to work on the areas that are linked to marine biotech, aquaculture, fish health, etc., etc. So one of the things that we need to focus on is to look at crop enhancement through genetic improvement. And the technique that people are using right now is the same CRISPR technique which we used in zebrafish
Nice.
to develop this mutant.
So you guys have
There is
adapted to
already
CRISPR
there is a
already.
connection over there. Yeah.
Mm-hmm.
So we might look at this particular gene in other fish and see whether it is indeed happening the same in other species. And if it does, then it can potentially get rid of hormonal treatment.
Right.
Because hormonal treatment would potentially mean that hormone residues being present in the fish meat, which
Right.
is when consumed by human beings, could have long-term deleterious
Right.
effects.
And it is also a continuous approach. Like you have to
Keep on
hormonally
doing that.
induce.
Exactly,
But
yes,
with the genetic, it is just you have to maintain a culture and it
And
keeps-
you produce that embryo in the lab
Right.
and release it into the
Right.
pond.
Right.
And,
Right.
and
You just
assure
have to
that-
make sure it doesn't get into the wild.
Yes, of course, that needs to be
Right.
taken care of. Yeah, but even if it goes into the wild,
Mm-hmm.
it is a fertile fish. It's not infertile, so it is not going to cause infertility, or it will not stop
But
the
it-
production. But
right.
it is as a male fish. So
But
in
the
the wild,
distribution of female and male may imbalance, like
skew,
75
yeah.
to 25%
Correct.
mathematically.
It could happen, yeah.
Right, right.
but then potentially there is no
Right,
danger.
right, right, right.
And it is not a genetically modified as well.
But yeah, there would be fight as well. If that happens, then there's a less supply of female and more oversupply of male. Then male have to fight each other for copulation, and then
But
there's
then,
a rebalance taking place.
But then
Mm-hmm.
when we are releasing, if accidentally it goes to the wild,
Mm-hmm.
the moment it goes to the wild, it will invariably mate with a normal female.
Right.
There is no homozygous.
Heterozygous there.
There is no homozygous female in the wild,
Right.
because only the males are homozygous. So if you look at Mendelian's law of distribution, if a homozygous animal mates with a heterozygous or a wild type, there is no way you can get a homozygous male again.
Right, right, right.
So yeah,
So when you said can be used as a biomarker,
yes,
like with my example where the fish turned green when the presence of estrogen. So do you recall any other example or possibilities of using zebrafish as biomarker which is contextually relevant to India?
people have used zebrafish as a model to study heavy metal toxicity, and the similar approach. So we have certain genes in the body that are used to metabolize heavy metals, any
Right.
chemicals for that matter. So these are metabolizing enzymes. So these enzymes carry their own promoter. So people have tried to get that promoter sequence upstream of GFP or RFP or whatever, and they have used it to identify. They've used it to understand whether the fish can actually serve as a biomarker for heavy metal pollution.
Wow, that means we can employ zebrafish in water reservoirs where
Yes.
we take the water from to the
Exactly.
municipality.
Potentially, you can
Yeah.
have, say, 1 liter of water. You release some zebrafish embryos and see whether they're turning red or green or yellow.
Right, right.
And if they are doing so, then depending on
Wow.
which promoter you have added,
Right.
you would also know which heavy metal it is.
Not
whether it
just
is
heavy metal, but also hormone or
anything,
other pollutants.
anything for that matter. Yes.
Wow.
Another example of zebrafish could be potentially, I don't know how many people are working on it, but they can be also used as bioreactors.
Means production of some desired chemicals through zebrafish. It's like
Proteins, enzymes,
Like
for example.
how?
For example, insulin. So
Presently, we use yeast
yeast
encoded with
or
insulin.
E. coli.
Right.
So basically, the same thing can be done. You can potentially make zebrafish produce a human protein which can be purified and used for whatever applications you
Oh,
want.
right. The proteins which can't be can't be put into yeast.
Not necessarily. You can, but
Why
then
prefer zebrafish
because it's
over
a big animal,
Right.
the amount of protein that is produced is also going to be more, and the fact that zebrafish produces so much of embryos, so much of eggs, and if all of them are fertilized, the amount of protein that you will get from one batch would be much more than what you would get from probably 10 such experiments in a E. coli or a yeast model.
Do you have any examples of like proteins that are produced?
People have worked on it. In future, we do plan to work on this, but at this point, we have not yet started. But we do hope that someone will start this. Yeah, a lot of proteins, a lot of enzymes, which are important for in vitro replication of DNA. There is a very important protein which is a polymerase enzyme.
Right, PCR enzymes.
In PCR, so that's a protein. This enzyme is obtained from a bacteria,
right?
Which is
thermophilic. Right.
Thermus aquaticus is the bacteria that produces that enzyme. Potentially, you can make zebrafish produce that enzyme.
Nice.
So
and
I'm just giving
the
an
cost
example.
of a PCR, possibly.
And then it can be used for PCR, and you can produce in bulk. You can have most likely it will have the same properties.
Nice.
There is no reason for us to believe that it will go different if it is produced in fish.
Yeah. So we covered disease modeling, environmental aspect. Do you have anything to say in drug discovery in detail? Like how do you
drug discovery,
model drug discovery?
I'll tell you about the basics of drug discovery. And the drug discovery is not a very easy thing to do. There are two ways. One is phenotype driven drug discovery. The other one is target driven drug discovery.
Okay, I can guess the phenotype driven, like changes in the morphology.
Correct. So you look at the phenotype, whether a particular drug when you're giving, whether the phenotype is changing for good.
Right.
The other one is target driven.
What is that?
Target means you're not looking at a phenotype, you're looking at the protein, a particular protein inside the body. That protein is there in some cell, but you're targeting that protein. So target driven is actually a faster process compared to phenotype driven drug discovery.
Right.
Most of either regardless of whether you follow a phenotype driven or a target driven, ideally a drug discovery takes about 8 to 10 years minimum.
Right.
Because you need to have different steps.
That too, if you're lucky.
Yeah. So zebrafish or any other model system will be used for the purpose of preclinical studies. When you say preclinical studies, it means the studies that you're doing before you take it to the clinics.
Right. Drug testing.
Drug testing. So you are looking at them at model systems, looking at the efficacy, looking at the viability, bioavailability, looking at its stability,
side effects,
etc. And then once you are confirmed and once you are convinced, then you take it to the clinical trials. And once it is done in clinical trials, then it goes for an FDA approval, etc. So it takes time. Now, zebrafish's role in drug discovery would be, as I said, to complement the existing model systems and to hasten the process of drug discovery, simply because they are amenable to high throughput screen, and because they are vertebrate model systems, they can be used to for the initial screens, certainly, and whatever qualifies for the next level can be taken for the higher organisms like rat or mice or any other mammalian system, or even higher like hamster or a chicken or.
But it usually starts with the computer, right? Computer modeling, and then it comes to zebrafish.
So when you are talking about the target-driven drug discovery, it is always with a computer model, because you need to have the structure of the protein, and you need to have the drug, and there is something called docking. It's an in silico simulation.
Compatibility.
Yes, whether the drug that you're discovering is actually able to bind to your protein of interest.
Right.
So that is compatibility, and whether if it does bind, how it changes the surface properties of that protein, whether it is inhibiting it or whether it is enhancing it, etc. Yeah.
Right. So now I'm interested in what's cooking in the Nitte at your college.
All right.
So what have you guys been working on? How many projects? What diseases? Everything in detail.
This institute started in 2013.
Okay.
As the name suggests, it's a research intensive institute. Nitte actually has been traditionally teaching oriented organization. They were mainly into academics, academic programs and they have been very successful in the academic programs that are being rendered by the various other colleges. But of late, because of the need for a research component in every academic program that you offer, obviously Nitte also realized that there is a very imminent need of investing in research, and not just research, quality research, because to be competitive, you need to be at that level. Otherwise, you are not there in the picture. So, with that background, 2013, this institution was started, and it was named Nitte University Centre for Science Education and Research. So, this is in something similar to what you see in the ICERs that we have by the Government of India. These are autonomous.
Equivalent.
These are called Institutes of Eminence by the Government of India. There also it is science education and research. So the faculty are primarily researchers who are also involved in imparting education that is related to science and research.
Right.
So you are
Discovering
developing
new knowledge, plus
imparting that, and you are generating skilled manpower who can contribute to R&D in the country.
Right.
So that was
Very
the background.
Much needed.
Yeah. So we started in 2013 in a very small way in the medical college. Last year we completed 10 years of our existence.
Congratulations. And you were there since the beginning of the institute?
Not exactly since the beginning, but I joined in 2014.
Okay,
but then I have been part of the
foundation
journey right from the formative years. Yes. So I have seen that institution grow. I have kind of involved myself in the growth right from the beginning. So yeah, I could say that I have been there right from the beginning. Yeah. So we started in 2013, and then quickly the management realized that this is a great potential and this is something that needs to be encouraged. So we moved to this campus. We were given some space in the pharmacy college of the university. 3 floors were given to us, and we started there. So 2015 we moved there, and 2013 NUCSER started as a biomedical institute
in combination with medical institute.
Yes, because it was the medical college already had a research facility, so they wanted to start something related, an allied health course in biomedical. And then we started with one undergrad program and one post-graduation program, and then a bunch of people came in, and a few researchers joined, and then we moved to this facility, and we started full-fledged research only from 2017. But the growth has been phenomenal. The pace at which we grew was really, I would say it is like at a lightning speed, because for a private institution to grow at that pace is something which is very surprising.
And also the fact that you guys have introduced cancer research for the bachelors.
Yes. And the good thing that we did was we brought in people who are involved in contemporary research. So that was important. So, and we brought in people who are active researchers themselves. And we always wanted young people to join, who are hungry for more, who are dedicated, who are passionate about their research. So we started bringing in people, and we started working on different dimensions, encompassing the various factors that influence human health. Right. So there is a concept called One Health. One Health means cross connection between different verticals that put together decide how human health is going to be.
So what are the verticals?
So for example, if we live in a good environment, when you say good environment, we need to have good soil, we need to have good water, we need to have good plants,
air quality.
So if we need to have all of these, so we need to know the soil health, we need to have an idea of the microbial health, we need to have an idea of the plant health, we need to have an idea of the animal health. So all put together decide human health. And so this concept is called One Health. So we started with this concept, where we try to connect human diseases with the factors that influence the likelihood of a human being getting infected by an infectious agent, which is directly linked to the water health. Because if the water is not clean, if the water is not sanitized. You do not think of having a system where you are not exposed to it, right? If it is unhygienic, you are exposed to it. Similarly, something that comes from the soil, which can get accumulated into the plants, which can then get into the animals, and then these animals are consumed. One good example would be antibiotic resistance, which is a big concern these days, because people potentially could die from a simple cough or cold if they end up contracting a bacteria which is resistant to all antibiotics. And this is primarily a big concern because we are all exposed to very high amount of antibiotic residues which are used in poultry and other meat industry. So, and by nature, bacteria develops resistance over a period of time. Yes, to evolve. Yeah, exactly. So, there are hardly any new antibiotics that have come in the last 100 years. People are using them in different combinations, etc.
Over and over.
So, indiscriminate use of antibiotics have led to this antibiotic resistance. So, people are looking at different other ways of biocontrol, etc. So, in this institution, our focus is on One Health. We try to connect
ecological aspect,
exactly, nutritional aspects. We talk about the environment health. We talk about the basic molecular biology and basic infectious biology,
direct and indirect.
Exactly. So we have 5 departments right now: Department of Infectious Diseases and Microbial Genomics. Then we have Department of Molecular Genetics and Cancer. We have Department of Food Safety and Nutrition. We have Department of Environmental Health and Toxicology. And then we have Department of Bio and Nanotechnology.
So all of them are using Zebrafish for their work?
Most of the people are using this in some way or the other. Primary users are from the Department of Molecular Genetics and Cancer, but we have a nice synergy between the
staff,
researchers. Because the model is such versatile, it can find application in any kind of research that you can do. I'll give you an example. I talked about antibiotic resistance. So people are looking at different other ways by which a bacteria can be managed. So one aspect is use of probiotics. Probiotics are good bacteria that have the ability to inhibit pathogenic bacteria, harmful bacteria. So we did a study on probiotic bacteria, and we use Zebrafish as a model to see in real time when you infect zebrafish with a pathogenic bacteria and you add a probiotic bacteria.
It was in the gut or on the skin?
In the gut. So basically you tag the probiotic bacteria with one fluorescent protein, you tag the pathogenic bacteria with another fluorescent protein. So one may be green, the other could be red. So when the red signal is there, it indicates that there's that the pathogenic bacteria is growing. And then you give probiotic bacteria in the same fish with feed or through the water. And you see gradually the red signal is diminishing, whereas the green signal is increasing. So that means at real time you're able to show that the probiotic bacteria is not only successful in colonizing the gut, but is also successful eradicating the pathogenic bacteria.
Effective. Create alternative against antibiotics.
So this work we have done on fish. So we published quite a few papers on that. And we have a lot of focus on the university has a mandate on the sustainable development goals that are laid down by United Nations. And every year the Times Group in the UK, they evaluate universities based on their activities outside education in the various goals that we are supposed to meet in order to become a sustainable earth in the next 20 years or 30 years, whatever. So there are about 17 goals that they have envisaged, and by 2030 we are supposed to achieve those goals to become sustainable. And as universities, we are being evaluated for what we are doing for those goals, how we are reaching out to the society, what kind of outreach we are doing, what kind of capacity building we are doing, what kind of research we are indulging in, what kind of innovation we are bringing in. So we are participating through our research, and one of the goals is life below water, which is how to protect the life below water. That means the fish and other aquatic resources.
So you mean how life is adapting to ocean acidity or something?
No, what you are doing to have a sustainable life below water. How you are not making the coral reefs go extinct.
Right.
How you are maintaining the biodiversity. How you are protecting the life or the diversity of certain endangered species, etc. So our institution is focused on the activities related to that. And last year, not last year, 2024, this year, we are 66th in the entire world.
Congratulations.
And we are number 1 in the country under life below water. Based
so
on
what specific researches have you guys done on life underwater?
So there are this example that I gave using the Zebrafish model system that we have done. There are so many other work that we have done on aquatic microorganisms, looking at the toxicological aspects, etc. etc.
All of them using Zebrafish as the model?
Most of them we use Zebrafish as a model system. And we have done different infectious studies, etc. We have looked at seafoods. We have looked at various aquatic resources. We also did quite a bit on developing functional foods from aquatic plants.
So what do you mean by functional foods?
Functional foods means nutraceuticals, incorporating some active ingredients which are present in marine organisms and marine environment, bringing them into their food and making a formulation, etc. So these have fetched us this recognition of being number 1 in the country, and also the icing on the cake is the Center of Excellence that is given by the government of Karnataka to work on marine biotech and marine innovation. And there's huge potential on the marine resources. It's not just a food resource.
Yeah.
It is the energy resource and the mineral resource.
Right.
These two are equally important.
I was equally curious when I was a bachelor student.
Exactly. Because, so an institute like this enables us to valorize these resources, to make use of these resources, to make a product better, to make a technology better, to come up with new products,
to discover new knowledge,
to discover new knowledge, and to have new drugs from the sea. In fact, you'll be surprised to know that almost 25 to 30% of anti-cancer drugs are from natural origin.
Right.
Natural origin means the active ingredient was first identified in some plants or some fungus or some marine organism.
That's like PCR protein.
So people have identified it, they have understood the structure of it, and then they started synthesizing it in the lab. But original discovery was from the nature.
Right.
So from marine organisms, there are plenty of examples.
Right.
One of the most widely used product from marine source is the green fluorescence protein, which is obtained from jellyfish.
Which was it? Oh, jellyfish, right.
And it is the most important protein in cancer research today. So
that has
there is hardly any cancer lab
without that protein.
Without GFP.
And to add on to that, like 99% of bacteria we don't know that is present in the ocean bed or sea bed or in the ocean.
Correct.
Just like PCO was found in the bacteria, the thermophil.
Right.
There maybe we're missing lots of proteins out there which are undiscovered yet to discover.
Yes.
Great potential.
So, institute like ours is primarily research oriented. Our focus is on research, but at the same time, we also look at teaching to generate skilled manpower. In the last 6 to 7 years, we have managed to get about 57 externally funded projects worth about 18 crores Indian rupees. And almost every faculty here has been the principal investigator of a project. So we have got small projects to medium-sized projects. We have been applying for infrastructure, but this COE is the first big infrastructure project that we got. The funding is for 7.4 crores for 3 years, and that includes funding for manpower, funding for consumables, funding for equipment. We are getting 3 sophisticated equipment under this new project, and things are looking good for us.
What is the new equipment that has come to the campus?
We are in the first year. We are going to buy 2 equipment. One is a confocal microscope, which is a very specialized very sensitive microscope which can give you ideas at a 3D level in a cell.
What was the magnification level of that?
Now you can go up to several hundred folds magnification. It's a very integral part of the set of equipment that you need in institutions, particularly those that use model systems.
Right, because in the
Zebrafish, or because you need to, it enables you to do, it enables you to do sectioning at in the Z scale. So we have in a 3D. So you have 2D would be X and Y, but then 3D would be X, Y, and Z. So confocal microscopes allows you to get section. So potentially you can see a protein which is at the surface of a cell to a protein which is deep inside the cell, which you do not see in a regular 2D system. So this is one, and the other one is a spectroscopy instrument which allows you to identify the composition of peptides or some active ingredients at a great sensitivity. Yes. So it's called the liquid chromatography mass spectrometry unit. So these are the two equipment that are going to come in the first year, and then third year we'll see the other things. Yeah.
So what are the core expertise that you guys have built at this institute in Zebrafish that enable in India, that nobody has worked on that aspect on Zebrafish model as you guys in India, that are in the world, that people can rely on you guys or come here for collaboration or to get the expertise from you?
Because it's a very competitive world, I don't think it would be appropriate on my part to say that we are the only people working it, because some people somewhere might be working on such techniques. But one thing that we are quite unique about it here in fish is perhaps looking at the use of the zebrafish as an infection model to study the effect of probiotic bacteria on pathogenic bacteria. This is something which is quite unique. Not many people have done. So I think this is one area that we are quite confident about. Yeah. Yes, that is something. And the other one would be by virtue of being a group that has a lot of people from an infectious biology background. So we have about almost 8 to 9 people in the group who come from that background. So this is something that is quite unique for us. So with zebrafish, I think this is something that comes to my mind at this point. But potentially bioreactors could be something that we are
planning next.
Which I don't think many institutes in the country are even looking at.
You're trying to stay one line ahead. So what are the other ideas you guys have for the future using zebrafish apart from bioreactors?
There's a concept called single cell omics. So because cancer is a very heterogeneous condition.
Right.
So there are a lot of heterogeneity between the cells in a tumor.
Right.
So there is a concept called single cell omics.
That means you study
individual
cancer
cell.
In a unit cell.
Individual cell. So zebrafish could be a potential model to study single cell omics.
How come it is multicellular?
Yes. So it depends on the tissue that you're looking at. So there is a disease. We have started this not with cancer, but there is a lot of relevance to cancer. But then there is a disease called disorders of sexual development. So patients have morphological manifestation of both the sexes.
But is it a chromosomal disorder?
No, it is a genetic condition.
Okay.
It is it's not at a chromosomal level. It's
like extra count of a chromosome, like we have in the Klinefelter syndrome.
Not a Down syndrome, not a Klinefelter. It is at a genetic level.
Right.
So, single cell omics is something that is being pursued as something that is supposed to have some clues on understanding how these happen. So, and in cancer biology, single cell omics is something very relevant because there's a concept of cancer stem cells. Because we say cancer is a disease that comes back. So the reason why I say this is because not all cells die because of a cancer treatment. Some cells become inherently resistant to chemotherapy or radiation therapy. They do not die. So these cells develop resistance, and the mechanism of resistance to study, it is very essential that you understand what is happening in a single cell. Why is this important? Because cancer originates from a single cell.
Right.
It is one cell going rogue in the body, leading to a catastrophe.
Right.
So
you mean it's better to understand the mechanism and process happening in one cell, later go to the sophistication and complexity.
So people are looking at how each cancer cell behaves because of the heterogeneity, and whether there is something that can be picked up, which is not common in other cells. And if that is known, then there could be a chance of developing a therapy which can target that particular cell.
Right. Or metabolic pathway in that cell.
Something like that, yeah.
So how do you induce a single cell matrix into zebrafish? Like you said,
so what we
specific.
intend to do is, we can use zebrafish because they are, transparent in nature. So two things can be done. You can use a zebrafish cancer model. You develop a genetically cancer-prone system in fish, and you use different categories of drugs and see which drug is most effective in killing the cancer cells which are glowing in the zebrafish. So that will give an idea to the clinician or the oncologist. Even before he or she is administering a drug, he or she would know which drug is likely to have the best effect. Other way would be develop zebrafish cancer model.
Wow.
The other part is develop a zebrafish cancer model, give one particular kind of a drug, and observe it for some time. So when you give a particular kind of a drug, it is understood that some cells will die and some cells will not. So the cells which are not dying, they are the resistant cells. Harvest those resistant cells from the fish, grow them in vitro, try to understand why they are becoming resistant. So if you know that, then potentially one can identify another drug which can block that protein which is rendering the resistance to that.
Wow, you mean this can be applied in real-time cancer treatment?
Yes.
Like real-time? Like
Yes,
suppose
it can be.
I'm a
Potentially
doctor
it can be, yes.
and a
We are
patient
actually
has
doing
come.
this. We are actually doing this,
Wow.
not with fish.
Wow.
So there is something called liquid biopsy technique,
Right.
where we analyze the presence or absence of certain mutations in patients to know whether they are likely to be responding to a drug or not.
Right. I mean, this deserves another podcast episode, liquid biopsy.
We can talk about liquid biopsy
Right,
some other time.
right.
Yeah.
But
That
I
we are
was
doing
thinking,
routinely.
like, I was just imagining when you said, like, as I'm a doctor and some patient is coming up.
I'll give you a brief. A patient walks into a hospital in the morning at 9 o'clock, goes into the OPD, gets checked, unfortunately gets diagnosed as a cancer or whatever. Or it may be a second opinion. He or she would have been diagnosed with cancer somewhere else. The person is coming for a second opinion, meets the medical oncologist. So medical oncologist says,
Sequences the DNA.
medical oncologist immediately sends about 5 ml of blood of that patient to a lab like ours. We extract something called cell-free DNA out of it, run a PCR, and look for the load of a particular mutation in that patient. In 2 to 3 hours, we'll be able to tell the
Abnormalities, which genetics.
we'll be able to tell the clinician or the oncologist whether this particular patient will respond to a particular drug or not respond to a particular drug. So by the time the patient is done with the routine examination of the hospital, the doctor is already aware of which drug is likely to be effective in the patient.
Right.
So even before you start the treatment, you are aware of the chances of the drug acting on that patient, because you're looking at the genetic makeup of the individual. So that is the power of...
Okay. I thought the zebrafish is involved
Potentially,
here. Okay.
no, in this we are not using zebrafish, but potentially the same thing can be done when I told you the example.
Right.
We can give zebrafish
Yeah, the same
a
sequence.
battery of drugs,
Right.
and then you see which is the best one.
Yeah.
And you can tell the doctor, these are all these drugs are good, but I think if you use this, there's a likelihood that this drug is going to be most effective.
Right. If it's a cancer thing on screening on the zebrafish, what indicators would you look for? Whether the cardiac, whether it's neurological in the zebrafish?
It depends on the kind of condition.
Okay, right.
In cancer, there is a set of things that you look at. In cardiac condition, there's a set of other things that you look at. In neurological, you look at the behavioral pattern, the function of the neurons, etc.
Do you see a commercial possibility for your institute? Yeah, because you guys have a zebrafish facility already.
Potentially, yes. Because now that we have a COE from Government of Karnataka, incubation and incubating startups is one of the mandates. So potentially, we see people coming up with such
new ideas
and
connecting oncology, patient-facing, patient-facing institute to the R&D like yours.
Right. That's a good chance. Yes.
Oh, nice. So you guys have any questions? Okay.
I think we're done.
Yeah, I think we're done. I think I want to badly see your fish facility and walk me through it.
Yeah. So they are all our students. Kritika they're all PhD students at different stages of their PhD. Kritika is also looking after the facility as a supervisor. She's appointed by the university. Nidhi is working on a rare genetic condition. Mrinali is working on something quite interesting. I think in the discussion didn't come up. Using bacteria as an anti-cancer agent. If you can whether you can use bacteria to treat cancer. So she's trying to understand this with zebrafish. And Sujita is working on the story that I told you about the male fish coming up, the homozygous one running up into males.
She was the one who discovered that.
She is the second one because the story was interesting, so we wanted to do a little more on this. So the baton has been passed to her and she is supposed to work on this. And there are quite a few people, but they have left for the day. So this is how the system works. I will let this guy speak.
How do you use zebrafish?
So I'm using zebrafish as a model system to study the tuberculosis disease. So I'm using Mycobacterium marinum bacteria, which is a natural pathogen to zebrafish. So I'm using it to infect the fish and to check the pathogenesis in zebrafish. I'm actually generating a mutant for a rare genetic disorder, specifically for diamond blackfin anemia, using zebrafish, especially on a transgenic background, where the blood cells fluoresce in green color. So you can see the entire blood cell of the organism in green. So wherever there's blood flow in the organism, you can see it in green color. So there's reduction or there's an increase in the blood cells. You can just find out without killing an organism visually. So
if you had to explain this work in simple terms to your parents, how would you say?
So basically, I'll be editing one of the gene which is responsible for the disease, and checking what would it do for the zebrafish, like how it happens in the human body. The same, what changes does it do in a zebrafish, and using that phenotypic or genotypic characteristic, you can find a drug or a natural agent to reduce or increase the ability of the
It's great that your parents understand genotype and phenotype. Just kidding. Yeah, what do you think?
Well, as Sir has already told, my work is mainly based on an alternative treatment for cancer. So as we all know that nowadays there's a lot of side effects that come with cancer treatment. So this is an alternative, that is the bacteria. It shows a potential to go and target the tumors. So my work might help in finding a different way wherein the side effect, the window of side effect also will be smaller, and the cost will also come down. So that is what my work is.
That's interesting. How about you?
So, as Sir has already told about how Pan gave rise to most of the meals, so my work will be the continuation to figure out Pan's role in telomeres. So, telomeres role in Pan. So, I will be doing in-depth study of this using Zebrafish as my model system.
They were born on the 25th of this month. So they are about, today is 29. They're about 4 days old now. 25th, so 26, 27, 28, 29. They are 4 days old. So now, as I told you, after 2 or 3 days they come out and they start moving around. So this will be very difficult to catch them unless you anesthetize them. And this stage, they are looking for feed. So that's why they are swimming.
What do you guys feed for this at this stage?
At this stage, we give them, not yet. Once they cross 7 days, we start feeding them with paramecia, which is again a live feed which is given. And the adults are given what you are seeing over there brewing. That is actually a cyst of a shrimp. It's called brine shrimp. It grows in very high salt concentration. So when you put them in very high salt, the cyst opens up and it germinates. So that is kind of an insect. So when it is given to this fish, it keeps moving and then they find it very happy to chase and eat. So this is about 4 days old, and you see another one here. You see here, they are swimming again. You see the babies.
So to capture that, you first have to put anesthetic agent and then you can...
Right. Now, so these are born on the 15th of July. So they're about 14 days. Now, they are good because, as I said, most of them will be somewhere in the middle of the water. So they are happily there. And if you see, the belly looks good. I mean, there's a kind of a small thickening part of the belly, so they look like they're eating whatever we're giving.
How do you guys observe it? It's so transparent, so small.
So as I said, you need a good microscope and loads and loads of patience.
So you don't have to dissect them, it's just transparent. You can just study it from looking from the outside of
till they remain transparent. At some point of time they will lose their transparency.
This is aesthetically pleasing. Right. So tell me of it.
And it costs a lot to maintain as well. Not only this comes from Italy. It's an imported system. It is from a company called Techniplast. They are the pioneers in this field. Most of the institutes in the country have this system. There are other players also in the field, but these guys are the most most widely recognized and the best one, I would say. Yeah.
So what this does, disinfects water.
This is called water treatment unit. So this is the head of the entire system. So this is the most critical part of the system. So this unit can support 5 such units. So right now we have only 2, but then potentially it can support 5 such units. And then what happens here, it's a complete recirculatory system. So the water that goes in is coming out and getting getting treated and going back into the system. And this is happening day in, day out. The water that comes in has to be an RO water, ultra clean water that goes in. So this water is already clean and then it goes inside. Then it undergoes 3 rounds of filtration. And then it undergoes an UV treatment, and then it flows into the system like through this. You can see here, that's the tap. When you want the water flow to be stopped, you can just lift it up. It stops. So when you have to feed them, you have to stop the water. Otherwise, the fish will not be able to catch them. So once it goes in there, the excess water flows back into the system through this conduit. It comes back into here. It undergoes a round of filtration, which is just a sponge filter, and then whatever ammonia is there, it is taken care of by some bacterial action, and then the clean water comes out, and this goes into circulation again. So, and as I said, it requires routine checking every day, every 2 to 3 hours you need to check whether they are fine, whether the feed is given properly. If there is excess feed, that also goes into this and it comes out. So if you feed too much, you are spoiling the water quality again. So it's a continuous process. So quite labor intensive, quite demanding, but then the pleasure that you get after doing a research, not necessarily in terms of the papers, but being a part of a system like this itself is a kind of a thing to be proud of.